What are you playing?
Sorry?
What sport?
I'm going to be cross-accrediting, but I'm
You're
doing
what?
cross-accrediting, so I'm going to be doing two sports.
Oh, right.
But I'm at the moment in the cheerleading team. So
Can't wait to see you in a short dress.
We won gold last year.
Oh, cool. Okay, the green light's on. That means they're recording all of your dulcet tones and mine. There are a few slides to get through, unfortunately. And if you looked at them before, you'll know some of them are a bit busy, but we'll get through them nice and steady. Just remind you during the break that not only is PASS operating, particularly live on this campus, but there's also that Boost program. If you looked on Views, they're going to give you $1,000 if you sign up for it. I'd encourage you to do that. It's actually a federal government-funded project, and I'll talk about that during the break. So today I want to talk about amino acid metabolism, and we start on the grunt biochemistry. Now, I kind of hinted at last week, biochemistry, particularly metabolism, is a very conceptual topic, and staying with it is kind of important. What I've done is pulled out some key bits from the textbook and from other areas to show you what is important. I've got a couple of animations in this unit, not too many, unfortunately. One today to give you an idea of how enzymes work, but I'm going to keep making hints all during lectures related to the lab activity and your reports, particularly strong hints on what things you should focus on in both of those activities. I showed you this circuit diagram last week. I mentioned that in there you might want to go to a web page. This one is really important for you, particularly as you head into your report, and that particular report is going to be talking about genetic disorders. And those genetic disorders, you'll need to get to key enzymes, key substrates, key products, cofactors. Where do you find that? In that web page. As we go through the next few weeks, I'll be dropping back to this web page. You should be going to it, and I'll mention it again in the Q&A tutorials. That web page maps all of this. It makes your life easier. So when you select your topic for the report, you just need to type in the keywords, go to this web page, and you will find where it is in this particular metabolic pathway. And you'll find all the relevant material, including relevant references. So if you don't use KEGG and don't have KEGG as a reference in your report, you might end up with a nice round number. That makes life easier for you. That's why I don't need you to memorize every pathway. Our job is to recognize who the key controllers are and how systems generally work. What are the key concepts? The things to keep in mind, because that software doesn't tell you all of the controlling mechanisms. It just tells you enzyme takes that substrate, turns it into a product with that cofactor. They're the genes, they're the mutations, these are disorders. It doesn't tell you how to manage the system. That's what we have to do. I mentioned before the key pathway down was carbohydrates. You had to look at that last week, just where you're saying this glycolytic pathway down. Back up is gluconeogenesis. Just jot down, we start with a 6-carbon sugar here. Down the bottom, just before we get to the citric acid cycle, just there, we end up with a 3-carbon molecule called pyruvate. P-Y-R-U-V-A-T-E, pyruvate. I'm going to keep mentioning it. You'll see it a lot today. I won't go into the details until later in the semester, but we keep coming back to that process. Now, I mentioned before, last week, I drew those blue tran tracks around this pathway and said this is inside the cell. There are many things that interfere with the cell that tell it to do things. But inside the cell, there are genes, activations, transcription factors that say how much protein, how much work gets done, who does what, where, and when, depending on your developmental phase. Those things we must keep in mind. And I point out over here that these cells can switch. They do different things depending on things like extremes in your activity. Not enough substrate, you don't work. Too much substrate, you store fat. If you're really young, you've got a growth phase. If you're repairing tissue, you've got a repair phase. If you're pregnant, you've got a pregnancy phase. As you age, you become senescent, you lose muscle mass, you lose protein. All of these things are critical for us. But as we'll see later in the semester, the last half, we're going to see hormonal regulation. Insulin controlling blood glucose, but not just that. Glucagon controlling blood glucose, but not just that. Epinephrine, you know what epinephrine's other name is? Adrenaline, write it down. We'll switch between the two. I don't care what you call it, but it is a small, very labile, unstable molecule that increases your heart rate and changes metabolism very quickly, as you would expect it to do. The other key one is glucocorticoids. You know it mostly as cortisol. We'll measure that later in the semester. The other one that's related is corticosterone. Cortisol, alcohol, corticosterone, ketone. Jot down, glucocorticoids are steroids, just like testosterone, estradiol, estrogen, they are steroids. What are steroids? Transcriptional regulators. They get right into the nucleus and regulate gene expression. And you're going to see how you can alter the cell at a cytoplasmic level fairly promptly or at the gene level. The key one I mentioned last week is this supply chain. Remember the analogy of the cars? How much have I got? What can I manufacture? How much the demand is? The key regulators I mentioned last week weren't the robots doing the work. It was who the foreman was, poor person, who the executive was saying, where's the input to regulate that pathway? That's this character. Underline it. The allosteric enzymes. You're not used to those yet, I suspect. Those enzymes get busy or get slow. They don't turn on and off so much, but they change their activity very promptly so that when the pathway needs to move quickly, they activate it. When it needs to move slowly, they slow it down, which is what your cells do on a daily basis. And think about it, this is in your body right now. This is what your cells are doing right now. Right now, you're looking at me, half going to sleep, it's 4 in the afternoon, and your cells are starting, oh, I'm tired, I'm hungry. Let me go home, and you've got 2 hours of this with me. We'll just count how many times we have to do it. It's not many. It'll all be over. And then, my goodness, we get an exam. But the whole time, let's think about what your metabolism is going through. We're going to look at induction and repression, changing those enzymes, how we do it. Commonly phosphorylation or dephosphorylation, but there's other events we're going to lead to and give you some key examples. We finish with this one. Yes. Thank you. Right, last semester you had to memorise all those, didn't you? They talk about the functional groups, their functional activities, which I've circled up there. I'll put boxes around. Let's have a look at them. Lysine, arginine, histidine. They're basic amino acids. Look at those nitrogen functions on there. Lots of N pluses there. The acidic ones, some acidic components hanging off, which means they'll do different things in your proteins. Keep that in mind. It's all up on views, except for the bit you have to write down. Keep that in mind for your protein assay that you're doing all semester. What is the red dye measuring? You're going to get these 20 amino acids in different concentrations. Does your red dye detect the arginines? Does it detect the amino function? Does it detect the acid function? Here's some others, asparagine and glutamine. They're kind of weird, aren't they? There's an extra nitrogen there with an O function. We've got the special amino acids. Why are they special? Well, look at cysteine. What's it got? Sulfur. What's sulfur's used for? Particularly sulfhydryls. Hopefully you did it last semester. Sorry? Disulfide bonds. You're going to see that in insulin. Insulin produces a number of disulfide bonds, 2 cysteines together. What does that do? It stabilises a protein, particularly an extracellular protein like insulin. Manufactured, a lot of cleavage, we'll see that later in the semester, sent out of the cell, running around your bloodstream, and it's got to be in a harsh environment, so it's got to be stable. Disulfide bonds stabilise proteins. They're not generally inside a cell, disulfide bonds, they're generally outside. Few exceptions. Glycine, pretty simple little molecule, isn't it? 1 carbon in that context. Proline, I mentioned last week, an unusual molecule. Why is it unusual? Proline can hinge. It makes proteins move around a lot. It can also be hydroxylated. All that stuff gluing you together, collagen, we talk about advanced cell, is proline hydroxylated, making you all kind of leathery, sticking together. Down the bottom here we see our hydrophobic ones. Whole slew, alanine, How many carbons in alanine? 3, keep that in mind. How many carbons in pyruvate? 3. If I took the nitrogen off alanine, I'd probably have pyruvate, wouldn't I? Can I do that? Well, who can help me manage those nitrogens? Let's look at these oval-shaped characters I pointed out. Let's look at glutamine. There it is, N, C double bond O, a linker to an N double bond O. Let's have a look at glutamic. What's missing compared to glutamine? The nitrogen. You're going to see that in the urea cycle. What's your first will assay? The urea cycle. So glutamine, in this case, we're going to see it later on, is going to be used to carry a nitrogen while you manage it. It can also be an amino acid to build something. So amino acids don't just build you as polypeptides and proteins, they can manage other things. Alanine's a carrier, we're going to see that later on today, to move your carbons back to the liver to be worked. I've circled sulfur down here, methionine. Why would I circle that? Is there any disulfide bonds in methionine? No. But that sulfur and methionine could be donated to make a cysteine. So if I've got methionine in my diet, I don't need to make as much cysteine or get it from my diet elsewhere. The red boxes. Do you recall last semester what those amino acids in the red boxes could do or have done to them? Sorry? Kind of, but not what I'm after. Thank you. Phosphorylation. You can phosphate those guys. Now let's have a look at those. Serine, threonine, tyrosine, particularly tyrosine, is phosphorylated. When it's phosphorylated, there's often a motif. A motif, 6, 7 or 8 amino acids linked to each other. We see that when you do bioinformatics or any gene work. That protein motif is unique, often a serine, threonine, tyrosine, a couple of odd little obscure amino acids, and then a proline. Okay, so I phosphorylate my tyrosine. What's my proline capable of doing? That as well. But what else can it do? It can hinge. So if I phosphorylate, something changes, which means now my protein, for example, could be inactive. I phosphorylate it, now it's activated. And vice versa. We're going to keep changing these proteins. They are moving in you all the time. That's all I care about with amino acids. You can go to a textbook and find their single letter, 3-letter code. I don't care. They're a molecular weight, but their functional activity is critical. What they do to you, how they manage you. Keep that in mind for your protein assay. You've got a red dye. Does it measure any of those amino acids? How does it measure them? The functional groups, their functional activity, what does it do? That's your challenge in the lab. Can I move to the next one? Okay. Before I do, just keep a little bit in mind, how complex and how simple some of these characters are. Some of these simple amino acid sequences you didn't think would be too hard to make. Some of these ring structures like tyrosine, tryptophan, phenylalanine, probably a little harder to make. That means we're going to have more steps to make them, which we'll see why some become important as essential and non-essential. We're ready to move to the next slide? Okay. I mentioned at the beginning of last week that I'm going to take you through ingestion, through the trachea, I should say the esophagus, down into your stomach, milling it around in there, into your intestines and absorption. What happens to get it into your cells? A few things to keep in mind. Many of you are medical science students, a lot of nutrition students, but even when you're eating now, think about what's going on to get those amino acids into your cells to do work, whatever that work is. Let's go through this process. There you are munching away. What's happening to that piece of bacon or steak, that bit of protein of tofu that you've thrown in your mouth? What's happening to that as it goes in your mouth to those polypeptides, to those proteins? What are you doing to them? Careful. In your mouth. How many times are you told to chew before you swallow? Usually lots. What are you doing chewing? Increasing surface area. You're breaking it up physically. Why do you cook things? To make it easier to break up and digest. But what are you doing to proteins in your mouth? Are you digesting those with any enzymes in your mouth? Who says yes? Who says no? Where's the no's? Oh, God, the yes has it. I'm afraid the yeses are wrong. The only enzyme you secrete in your saliva is amylase, which is breaking up carbohydrates. You are not breaking up polypeptides. That doesn't happen until it happens in your stomach, which is then going on to your intestine. That's what's happening here. You are breaking up physically those polypeptides into smaller fragments, but you don't break the peptide bonds, you're just making more surface area. You get down to your stomach, what pH is that normally? 2, 3, depending on what you've been eating, drinking. You then get into your stomach. Does anything happen to them there? No, not much. You'll acidify a few and do a few other things. Do you absorb those polypeptides or proteins into your blood or lymphatic in your stomach? No, that doesn't happen there either. What is your stomach? A big muscular organ. All it's doing is it's kneading and doing and kneading. Occasionally put it out, it comes back again. Back in you put it, then it's kneading and doing, and then it'll pass through. If it passes through, then we start the process of digestion and absorption, truly getting it into your circulation. A couple of key things to keep in mind. How much do you need? Well, depends on your age, your growth rate, what you're doing, repairing, making babies, whatever it is. The recommended daily allowance is around 40, 60 grams per day. Do you eat more than that or less than that? Depending on your body mass. What's the nutritional value? What's what we're trying to discover now with some of the research we're doing. Plants now, during our development of higher climate temperatures, are not putting as much protein into their plant mass. That's going to become an issue for you later on. For you, your problem is you do not store surplus proteins. You have to work, lay proteins down. The moment you stop working, physical activity, you will lose that protein mass, which becomes an issue for you. Because if you turn that protein into waste products, that's nitrogen, where do the carbons go? So I was going to tease you all semester. What do you do with those excess carbons? You turn them into fat. So you're all sitting there looking at me making fat, aren't you? The one thing humans are very good at is making fat from excess protein. You don't store protein. You turn it into excess fat and urea, which we're going to get to today. You turn them over, depending on your activity. Your breathing, so your intercostals are pretty firm, but then you might not use your abs like me, 12-pack. So you'll lose it. He wants to have a look. This is a family show. Okay. All right. So you've got to manage the nitrogen. So as soon as you take a nitrogen off amino acid, let's go back one slide. As soon as you take a nitrogen off any one of those guys, you have to manage. It's toxic. You've got to put it somewhere. Well, we just saw you could put it on our glutamine, couldn't you? Then run it around and manage it. So it's not toxic as ammonia, it's nitrogen on somebody. And I'd like you to think like that. Instead of a free molecule being dangerous, I put it somewhere and keep it safe. Move it around until I get it back off again. I compartmentalise it, basically. We're done with this one. Okay. Is everybody right? Okay, now that you're all making fat, let's start the process of breaking these peptides up. You've just masticated it. You've increased the surface area. You've then swallowed. Hopefully it stays in your stomach. There's no other issues there. Here we are in your stomach, very low pH, very muscular organ. You should know, you've all vomited, I assume, at some point. Things can come shooting out pretty vigorously, can't they? Do they talk about that in Intro to Human Phys? No, what's the first physiology unit called now? Sorry? Concepts of human. I used to teach Intro to Human Phys when people were away and loved talking about vomiting, because it makes you realise how much your physiology is so intricate. What's going on here? Well, in your stomach you have a pH between about 1.5 and 3.5. We said roughly 2, it depends. What happens in that stomach? Enzymes are inactivated. You're not breaking things up. So even that amylase that was in your saliva stops working in your stomach. It's just another protein. But in that stomach, we're starting to see something like pepsinogen, I'll talk about in a moment, turned into pepsin. It's starting to be activated. Pepsin. Let's go back a slide or two. Let's have a look here, these little arrows. They're linking amino acids. What's the bond between amino acids? What's it called? Peptide bond, thank you. A peptide bond. So pepsin breaks peptide bonds, starts to separate amino acids. Here we are doing just that. Once we've kind of mulled it around, made it all kind of like soupy, like minestrone, we then pass it down into our digestive system. This is when the work really starts. Really starts because of this thing, your pancreas. Your pancreas does a number of things. We're going to deal with insulin, glucagon and the like, but it also secretes zymogens. These are, write down, inactive enzymes. These are proteases or peptidases.
Which ones, right?
Zymogens are inactive peptidases or proteases. That is, they're going to cleave peptide bonds. But ask yourself, why does, in this case, your pancreas, when you start off with your gallbladder, make an inactive peptidase? Send it into your intestinal lumen. There it is, big tube. Pushes it in there, and then you activate it in the lumen. Why isn't it active out there in the cells and tissue? It'll break you up, won't it? Big problem. So what we do is make sure it's inactive here, send it into our duct, into our intestinal lumen, and then we activate it. Who activates it? Pepsin. Pepsin that was activated here helps activate all these zymogens. These zymogens, I'll show you a few of them soon, start cleaving peptide bonds. Let's go back one slide, and I keep doing this all semester. There's your piece of steak or bacon, or your tofu, depending on what you eat. You cannot absorb that through your gut epithelial cells. Talk a lot about those in advanced cell biology. They are really important cells. You can't absorb that, but you can absorb maybe one amino acid, two, maybe three at the most. A few exceptions. Here we've got to break them down with these enzymes. So we are fragmenting those polypeptides to be able to get them across your gut epithelial cells. Here's your gut epithelial cell. In advanced cell, we start down the bottom. That's the crypt. Stem cells, nice little stem cell niche down there in the bottom. The cells migrate to the top of that villus. 4 or 5 days, cell dies. You have a new gut cell every week. A new gut every week. Constant turnover. Stem cells down there are very delicate, but a very niche environment, controlling the process. So they're trying to absorb amino acids into your circulation. They're doing that as di- and tripeptides, and it goes into the blood. Please underline the blood. The amino acids go directly into the blood. We're going to come back to fat in a few weeks. The fat doesn't go into the blood. Fat's going to be more complex. We have to control it so it doesn't form lipid droplets in your bloodstream. All of that passage and the rate depends on what you eat, how active you are, how old you are. All of those things vary. Think about what you eat as a meal, how quickly it comes out. Now, I can't stress enough, much as it sounds rather disgusting, you should look at your stools. You know, the 3 Ps, the pee, the poo, and the paper. Look at your poo. Not every day, but regularly to make sure it's okay, there's no issues with it. When you get above 50, you'll get a notice in the mail, send us a poo stick sample and we'll check you've got colon cancer or some other issue. Because these cells turn over so rapidly, they're more likely to become tumours. All of us have polyps at some stage, but these cells are so important to you to try and absorb things. We'll talk about things like Crohn's disease and others later on, where they don't function correctly. They don't get the absorption across. We done with this one? No? So you're sitting there thinking, oh, my poor stomach. Next week I'll have a new one. Toxins cause all sorts of issues. Now, while we're here on this slide, just jot down, you do not absorb anything into your circulation in the stomach, except vitamin B12, except a few drugs. Some particularly designed to use vitamin B12 as a carrier to absorb those drugs into your circulation. There are a few toxins. that get into your stomach that can affect your stomach. But there's no absorption there really at all. It only occurs down here in your duct, the lower part of your gut. Can I move on now? Thank you. Everybody else right? Is that a yes? Okay. There's a piece of steak or that bacon I was talking about. There it is. Amino up the top, carboxy down the bottom. Let me go back a couple of slides. There it is, amino, carboxy. Same deal. What you're seeing here is peptide sequences. You're seeing the amino acids. What you're also seeing on this slide are the enzymes, the proteases, that break up that long chain piece of bacon polypeptide or tofu into fragments. Please look at the ends first. Don't write anything just yet. Look at the ends. Look at the top. What do you see there? An aminopeptidase. What does that aminopeptidase do? It looks for the end, the amino function, doesn't worry about the R group, and it will cleave that peptide bond where you see the arrow. Does that make sense? Yes or no? Indeed. So this aminopeptidase finds that amino. It doesn't care what the R group is. It could be proline, alanine, lysine, glycine, doesn't matter. Once it finds that free amino, it latches on and cleaves that bond just there, breaks that peptide bond. So you end up with a single amino acid, right? And whatever else the piece of steak is. Is everyone clear on that? Let's look down the bottom. Here we have a carboxypeptidase. What does it look for? The carboxy. It doesn't care what the R function is generally. It breaks that peptide bond. So let's ask ourselves, if we had an amino and a carboxypeptidase, here's a piece of steak, how long is it going to take us to break that down into individual amino acids and absorb it? 1, 2, 3, 4. Well, that's going to take a long time. So what have I got? Other amino acids. Now I need you to look carefully at this one for a start. Pepsin, where do we see pepsin? The stomach. What's it looking for? It's looking for the R group that's either phenylalanine, tyrosine, glutamine, aspartic. Doesn't matter which one of those it is, doesn't matter what's above it, up there, it doesn't matter what's below it, but if it finds any of those 4 amino acids, it cleaves that peptide bond. Is that kind of clear? Trypsin's another one. What does it look for? Arginine or lysine. Doesn't care what's above it or below it. As soon as it finds it, it cleaves that peptide bond. Elastase is down here, and I have one of my few animations for this one. Looking at chymotrypsin. What is chymotrypsin doing? Looking for phenylalanine, tyrosine, tryptophan, or leucine. Doesn't care what those amino acids are. Let's make that a little bit bigger. Now here's an enzyme. Now it's worth keeping this in mind for your assay, for your protein assay. You've got 20 amino acids. What does your protein recognise? Specific amino acids, sequences of amino acids, like this one's doing here. Let's go back to the start. What is that? You've seen all those ribbon functions before, haven't you? Beta sheets, helices, domains. That's a protein. That's an enzyme. It has an active site. You can see it there. It has a histidine function in there. Quite busy one. There it is. Let's push it off to the side. There's our chymotrypsin. What do we need? We need that piece of bacon with its long polypeptide. I don't care what's involved. Ooh, that was a bit silly. Let's get rid of that stupid thing. Let's get to that. Okay, let's draw the line there. This long blue thing here is just simply the polypeptide of the enzyme. Is that kind of clear? And of the enzyme, there's a serine there at position 195, and we keep on going, and there's another amino acid, histidine, at position 57. They come an active site. What do I need? My piece of steak. Why I've moved it off to the side, because this is chymotrypsin. It's looking for any of those 4 amino acids. Doesn't care what's up there, doesn't care what's down there. Those are the R functions on this piece of steak. Let me circle that. That's your piece of steak. There's my recognition domain. What's my active site in chymotrypsin going to do? Cleave the peptide bond. While it does it in 2 stages. So now I've cleaved it. There goes part of my steak off for someone else to have a go at, maybe an aminopeptidase, elastase, whoever. But for a moment or two, there's the other piece of my steak stuck to chymotrypsin covalently. Halfway through the reaction, what does the reaction need to do to get rid of that piece of steak off chymotrypsin? Cleave that last bond to our serine, separate. Now you've got 2 bits of steak, much smaller now, haven't you? When I do that, what do I achieve? Here's my long piece of steak. Chymotrypsin just cleaved it. What have I got now on either end? Apart from a thumb and a little finger. Absolutely. So I've got a free amino, free carboxy. What do I need? Amino, carboxy, peptidase. So now instead of going 1, 2, 3, 4, take forever, big piece of steak, fragment more, free aminos on the end. Is everyone clear on that process? Your stomach's doing that every time you eat a meal. We comfortable with that concept? Hearing aids going flat, you need to yell out. Can I move to the next one? Yes? Okay. Now we're down in your gut. Just look at the schematic for a moment. This pinky thing that looks like it's got a fuzzy hairdo is your gut cell. Let me go back a couple of slides. I keep doing this. Here it is. That's a villus. That's a bunch of cells lined up on this big long stalk. You're looking at the screen? There's a bunch of cells lined up on that stalk, but each cell has a funny hairdo. That's one cell. There's another cell. Each cell has villi, increasing surface area. So a villus, I go to the top, cell dies. Next one, cell dies. So now we're looking at the schematic. Oops, too fast. This is the individual cell with its villi. There's my villi. At that villi interface, I want to absorb the amino acids, better if they're individuals. So I've got a bunch of enzymes cutting up my peptides from my steak or my bacon or my tofu. There's the fragments. When I get close to the villi, I have my amino and carboxy peptidases, because when I cleave off that one amino acid, what do I want to do with it? Absorb it. I don't want to give it to some microbe, some fungi, bacteria, someone else to have fun with it. I want to absorb it. So your job is to have these carboxy and amino peptidases, or the dipeptidases, at this area called the brush border. So that when you cleave off the individual, you've got the chance to absorb it into your circulation. Your gut bugs are trying to do the same thing themselves. So your job to try and keep these enzymes close to that brush border. Now that happens for most of your life. This process where you are absorbing these things is called the absorptive state. You masticated, you digested, broke peptide bonds, that's the digestion process, now you're absorbing. Kind of clear on that simple, straightforward step? Good. Remember, the stomach doesn't absorb anything. All it's doing is just kneading and moving around, making sure the acid gets in there, trying to increase surface area and make it look like minestrone. So we're past that. This is down in your gut epithelium. Let's go back here, down in this region here of your gut. Is everyone clear on that bit? Good questions, clarifications. Okay, your amino acids, they're going to go into your blood circulation. They'll pass through the liver, who'll start to manage. Now, I want you to jot down now, the liver is like the major manager of most of your incoming substrates. It's trying to manage everything, including glucose levels. Insulin tells the liver what to do. And that's going to be based on what requirements you have. How much energy is ATP, how much work you do, what your body is going through. If we go up to the fats, we go upwards, you're going to absorb those. I hinted at last week, they're droplets. They'll want to coalesce. You've got to put them into a vehicle called a chylomicron from your gut. We'll see that in a couple of weeks. Then to the lymphatics. then into the circulation, they're going to have to be managed more carefully. Carbohydrates, what do you do with those? Well, it'll be kind of similar to the amino acids. Long polysaccharide, there's your mouth breaking a few up. Amylase gets to your stomach. Remember, it's a protein. What happens in your stomach? The acid neutralises it. No more breaking up. Get into your gut, we'll see in a couple of weeks, you start breaking it up again. You can't absorb long-chain polysaccharides. Glucose and fructose are what you absorb in this gut region. And again, your cells are trying to manage that into your blood. Your liver's managing how much you use, trying to constantly maintain your blood sugar levels. If not, you may well have a diabetic state coming at you. Except, and you'll notice I'm going to have a few of these exceptions during the semester, often in italics. How many of you had kids? Come into the world, there they are, lovely, patting them, blah, all over you. It's lovely. And if it's not at the end, it's the other end. But when they're first born, as mammals, what do they need to do as mammals? Sorry?
Milk.
They need to drink milk. They're mammals. So they are suckling at that point, and that point, up until about 4 years of age, their immune system is not well developed. Particularly in those first few days of their life on Earth, they need to get antibodies to protect them. Where do they get them from? The mother's milk. Now, an antibody is a big Y-shaped molecule. There's variants of it. Heavy chain, light chain, big domains, but it's a protein. It's a polypeptide with disulfide bonds. If you put that in your mouth, take it through your stomach and into your gut, and all of that stuff is there, all those peptidases, what happens to that protein? You'll fragment it. It won't not work any longer. So what does an infant do? Instead of going through all of this cleavage process, an infant's gut is not well developed. All of these cells are still working out what to do. In the first few days, your gut, as an infant, actually absorbs through pinocytosis. You know what pinocytosis is? Where you create little bubbles around the antibody and take them into the cell with your lipid bilayer. If you do advanced cell, we talk a lot about how you form those little bubbles. Take them in as whole antibodies, still functioning. The infant does that and then passes them into their circulation. That's how they protect themselves. They start to develop their immune system. As I said, it usually takes up to about age 4. But those first few days are critical. Without the maternal IgA, they often don't survive. But it's pinocytosis, so right down the exception to this process of gut digestion, absorption, is an infant, particularly mammals. Now that varies with different species. Can I move to the next one? Yes, ma'am? Antibodies are very large molecules. They're about 120 to 150 kilodaltons, so they're huge. They have heavy and light chains. So you can imagine me, there's my heavy chain. They have light chains, binding domains that grab onto the antigen. Those have to stay intact to work. But they are proteins, so just like that piece of steak or bacon, if I start cutting them up, they don't work anymore. So they have to stay functionally intact. Everyone else happy with that? So you've gone past your antibody stage. If you're going to have children, you'll see why you need to make sure they get their maternal antibodies from the mother. And anything that's like, you see that formula milk, that's often ultra high temperature. The antibodies are deformed, they don't work as well. So it's often best derived from the mother, not from some manufactured site, which we can talk about later on if you want. Okay, can I move to the next one? Thank you. Now we're in the circulation. Haven't talked about how to control it. We want to get it into cells. Which cells? Well, I said the key controller in your body is the liver. It's deciding incoming, outgoing, who needs what, where and when, but it's being controlled itself. We've just seen in this last few slides dietary protein come in. Assuming we're an adult, and we've all gone through puberty, haven't we? Yes? Okay, so we're all adults. There's your amino acids. They're floating around in your bloodstream. Who needs them? Well, whoever needs them might be cells of different types. Someone will grab them, but your liver is going to say, how much is in your circulation, what I'm going to do with it. If you don't use those amino acids to build protein, there it is, I've built it up. As soon as I start losing it, it goes back via the liver. The liver says, okay, there's excess amino acids. I might make some for myself, my own proteins. What does the liver make? Enzymes, proteins, more cells, depending on what it's doing. It might make some nitrogenous base compounds. I'll cover that in a moment. But if you've got plenty of those, the liver's all happy and healthy, it sees those amino acids as excess, not for requirement. What does it do to them? Well, it might well take them into removing the nitrogen, I should say. If, for example, you are on a high protein diet, you're not taking any carbohydrates, no glucose, it will turn some of those amino acids into glucose. High protein diet, you make glucose. High protein diet, you'll also start making fats. Now I'm going to use the term triacylglycerols, fats interchangeably, a long chain carbon. That's all it is, a long chain carbon. There are different types. Triacylglycerols, dare I start this? 1, 2, 3 long chain fats linked with a 3-carbon glycerol moiety. Those form your lipid bilayers around your cells, your nucleus, vesicles all over the place. They have a lifespan. They have to be turned over. You make them, you package them in your liver into what you know as VLDLs, very low density lipoproteins. Write it down. That's a vehicle to move them around so they don't form droplets. They don't damage your bloodstream unless you overwhelm it. Now, you're trying to manage the incoming proteins, the incoming amino acids, I should say. Excess carbon, I make glucose. As long as my blood is happy with glucose, my brain is happy, I keep my glucose levels constant. I can also send some of that glucose into storage. Glycogen is storage of glucose. Now, my colleagues at Hawkesbury are working on plants. I'm helping them with some advanced hyperspectral analysis because glucose traps water. It loves water. That's what plants do. They trap water. We're finding an unusual fluorophore trapping water. But your cells do it too. So every time you have a high glucose level in your blood, you'll have lots of water, which can actually make issues for you health-wise, we'll talk about. So to make sure that if you have a high glucose concentration inside a cell and the cell doesn't explode with water coming in being trapped by glucose, you store glucose, write it down, as a polymer. That polymer is glycogen. It's a branched chain polymer. Now think about that in a moment, I'll come back to it. There's my individual glucose molecule, loves water. Lots of water, cell explodes. But if I've got my glucose molecule linked to another one, it doesn't like water as much. It's now becoming a polymer. It's easier to manage, things like osmolarity. We're going to talk a lot about this process of absorptive state. In this case, we're talking about during the absorptive state. So keep in mind, you've just had a meal. Might be a few hours ago now. You've just had a meal. You've got lots of amino acids in your bloodstream, I should say. You're managing the process. Putting some things into storage, some things for utilisation, managing your blood glucose if you're not on a high glucose diet. This is the fed state, just underline it, the fed state. Your gut is managing stuff coming through. The fasted state is when you haven't eaten as much. Now let's go back a slide, just look at something. The last process lasts about 4 hours. Why did I put that up there? You wake up in the morning, do the usual stuff in the bathroom, come and get something to eat normally, don't you? When do you roughly have lunch? How many hours after breakfast, or whatever you call it, brunch? 4 or 5 hours, normal? You have lunch, roughly 1 o'clock. When do you get hungry again? Maybe a snack about 4 or 5 o'clock. Come home, raid the fridge. Hungry again, maybe about 6, a little bit. It's roughly 4 hours that your intestines, your circulation says it's time to go and get something to eat. We'll see your stomach manage you later in the semester. You are not in control. Oops. Are we done with this one? Not quite. Sorry?
I'm like a newborn. I need to eat every 2 hours.
Some people do. They have a high metabolic rate. We'll talk about that as well. Some people turn over a high metabolic rate. You'll see them. They eat all sorts of junk, high glucose diets, and they don't get fat. Everybody's different. Can I move to the next one now? Is that a yes for everybody else? Okay. Right.
Quick question, please. What's the LDL stand for?
VLDL stands for very low density lipoproteins. I'll bring you back to that when we talk about fats, because there are HDLs, LDLs, VLDLs, IDLs, and all sorts of Ls, including your P plates. Now, this slide I'd like you to keep in mind as we go through the whole of the semester and get to the exam. This slide is a nutshell version of this unit. It's encompassing, but it's missing things, missing key regulators you'll need to add to it as we go through the semester. As you can see, it's grey. Well, grey's back in fashion. That's a good thing, because I used this many years ago and I'm still using it. Thank goodness it's back in fashion, including the tight jeans and all the same stuff you guys are wearing now, I used to wear when I was your age. But this slide is critical because what we can see here is the intestine. We're seeing amino acids come into the bloodstream. You're going to see that switch to fats in a week or so, then to carbohydrates. What you're seeing here is the bloodstream, and this outline here, where my laser's running around, is the cytoplasmic membrane of a cell. Just jot down, particularly a liver cell, a hepatocyte. This liver cell is trying to manage glucose, fats, amino acid. What are the three major substrates we're managing this semester? The things you eat. Not so much drugs, but fats, amino acids, carbohydrates. Today and next week we're dealing with amino acids. Not enough seats up there? Yeah, I know, they're going to make it difficult. You have to move right along. Comfortable now? All set? Guys right? Cool. Let's get back to work. There are key pathways. Let's quickly go through some of them. On the left, glucose, down to pyruvate. What's that? Glycolysis. Back up, just a little arrow, put gluconeogenesis. We're not going to cover all the details now. When you store glucose, there's glycogen. That's the polymer I just talked about, isn't it? But that polymer's got to come back out ready to make glucose. What I want you to jot down, glycogen is stored in your liver and muscle. Not in your brain, liver and muscle. Particularly liver, so it's quick access. Muscle for quick access, for glucose. Fats are going to be difficult. Long chain fats, we're going to have to manage those. In the liver, we're going to have to run through this thing, and I want you to circle acetyl-CoA. Now, last week I showed you some key energy electron proton carriers, didn't I? NADH, ATP, FAD, and so on. I want you to think about acetyl-CoA, and you'll see its structure, and you'll have to know its structure, one of the few you have to know. It is a carrier, but it activates things. It's a bit like a long, chunky phosphate, and when it's on somebody, it's activated them to do some work. And that's what its role is. We're going to see it activate fats. We're going to see it activate fats to get into your blood, into cells, into your mitochondria. We're going to see it do other things as well, but it's an activator. I want you to think about acetyl-CoA as just a 2-carbon acetyl, and think of the CoA, draw a circle around it. CoA is the carrier. It's an ugly monster. CoA is the carrier, you're moving 2 carbons. Pyruvate is 3 carbons. That means if I'm going to make a 2-carbon acetyl-CoA, I've got to remove a carbon. We're going to do that when we start dealing with fats. What's this round thing down here? TCA, citric acid, Krebs, I don't care what you call it. In the middle, we're going to see the electron transport, oxidative phosphorylation. We've got plenty of time to get to those. You'll be bored before we get there. Are we there yet? What I've circled with those dashed lines in red is amino acids. They do a number of things. I'm hinting already that they need to get into cells. Here it is, going into cells. Do you need to break them up as individual amino acids to get them across the cell membrane? No. You did that in your intestine, didn't you? Broke them up into single amino acids, now they're in the circulation. You need to get them through the lipid bilayer. Well, now they're individual amino acids. Can you make proteins? Yeah. That's what ribosomes do, doesn't it? Translation. You're translating an mRNA sequence into a polypeptide. You're making the proteins. Then there's post-translational modification, all that stuff, you know, like making insulin. We'll see that later in the semester. I won't show this animation now. I'll come back to it. Those amino acids can be used to make other things, like these characters. Just write down nitrogenous-based molecules. Nitrogenous-based, nitrogen-based molecules. Some have different carbon sorts. We're going to talk about some of them. Heme's one of them. What does heme do for you? Makes hemoglobin, carries oxygen around. There's lots of nitrogen in there. Forms a chelate group. That's a critical molecule in red blood cells. Many others, some of those, melatonin, norepinephrine. Do you know what norepinephrine is? Noradrenaline. Those are nitrogen-based compounds. Those I want you to jot down as messengers. They are extracellular messengers. They're telling cells to do something. We're going to come back to those later in the semester as well. They can also be used to make purines and pyrimidines. Where do you find those? On your bases when you're making DNA and RNA. So you know amino acids can be used to make other things, but I want you to jot down, you generally don't make those nitrogenous bases, those nucleotides, because you ingest enough. It takes a long time to make them. There's lots of enzymic steps. I'll show you one later on. See creatine phosphate? Just underline it. You see the dashed line to creatine? Underline it. That's a carrier, a weird one. It's moving ATP energy from the mitochondria to the cytoplasm in your muscle cells. I'll come back to that later in the semester, but just underline it. It's a key carrier of phosphate inside a cell. What are you also seeing here? Well, the amino acids, if they're not being used to make proteins, there's a turnover. There it is. If I don't work, I lose it. When I lose it, what do I have to do? What's the end circling? Circle that. That's the nitrogen. What do I do with that nitrogen? Well, I might have to remove the nitrogen, called deamination. Deamination. I might, well, transaminate somebody else. I could use this character, alpha KG. There used to be a radio announcer in Adelaide called KG. Not particularly a fan of him. But I want you to write down ketoglutarate. Alpha ketoglutarate. Where do we start off with? Those amino acids. Who could carry a nitrogen as an amino acid? Glutamate, glutamine. What are you doing here? Inside the cell, you're putting a nitrogen on somebody so it's not toxic to the cell. While that nitrogen's on somebody, it's not dangerous. While it's not dangerous, I can decide what to do with it. I can either put it on somebody else and make some other amino acid, some other molecule, or what's down here? The urea cycle. Not this week, we're just going to give you a heads up on it. What are you doing? Well, you're seeing here these characters, glutamine, OA, and a few others, our nitrogen. What are you doing? Just keeping the nitrogen busy so it's not loose. You run into the urea cycle. Here it is running around through this thing, carbamyl phosphate. Now, one thing I'd like you to jot down on this slide as well is the notion that when you put a phosphate on somebody, you activate them. Not just turn the enzyme busy, but you activate them. Phosphate's high energy carrier. When it's on somebody, they can be activated to do something. We're going to see a bit of that today. The urea cycle, you see it running around a few characters. Right now you don't care who they are, you don't want to know, but some of them you will have to know. But what you do hopefully recognise is one amino acid, two amino acids, particularly arginine. It's part of the urea cycle. That'll be important for you as an infant growing through adolescence. As an adult, it's not important. Why is that? Well, it depends on whether it's an essential amino acid or non-essential. Depends on your demand. Now, this slide I'll keep popping up all semester. Each time I do, I'll highlight different things. Each time I do, every time I make a hint about something, I'd suggest you write it down. When you get to the final exam, and here's a strong hint for you, 4 short answer questions. Question at the top of the page, lines, a blank page behind, next one. I recommend that this thing, don't take it in, you know the trouble that'll cause, particularly the paperwork for me. But as soon as you get time to write, you sketch this thing out. Because it puts into perspective everything. You're going to add some key players to it. But if I see this sketched in the back of the exam paper, I know you've understood at least the basics of metabolism and who might be the major controllers. Because once you know this stuff, the rest is actually pretty straightforward. Comfortable with this one? I'll move to the next one. Okay. I just dropped a hint a moment ago. How many steps it takes to make amino acids? Well, if you ingest them, and particularly in this part of the world, is anybody on a nutritionally deprived diet? Generally no, unless you're on some weird diet. That's up to you. So you have a balanced diet, you shouldn't have too many issues. Well, what I'd like you to look at is this little graph down here on the left. It gives you an idea of why sometimes you ingest the things you do and sometimes you don't need to, depending on the state of your development. Down on the x-axis, what do we have? The number of biochemical steps to manufacture a particular amino acid. What you have here are the number of amino acids. So there's a number that can be made with just one step, 4 of them. One step, you've got an amino acid. What's one of those? Pyruvate, 3 carbons. I stick a nitrogen on, what have I just made? What's the 3 carbon amino acid? Starts with an A. Alanine, jeepers. Alanine. Take the nitrogen off, what have I made? Pyruvate. Can I make glucose from alanine? Yep. But you don't generally have to go through pyruvate. What about this one? We've got a few more steps, and as you can see, the number of blue, look at blue, what's blue? The number of non-essential amino acids. Why? Because if you don't ingest them, you can make them from somebody else. But as I increase the number of steps to manufacture, remember those complex ring structure amino acids, it's a bit more work. You don't have all the enzymes capable. You've lost some of those. Other species have them. So what do? You ingest them. What are they doing? Making more complex what we call the essential amino acids. There's the list. Non-essential, alanine, asparagine, aspartate, glutamate, serine. Hang on, serine. What can it do? It can be the base to make cysteine. What do I do to make cysteine? I need a sulfur. Here it is, coming from methionine. Well, we've got glutamine, glycine, proline, tyrosine. Well, there's tyrosine. It's kind of important, ring structure. But if you looked at its structure, it's not too different to phenylalanine. So I just make a modification, I can make tyrosine. Here's the essentials. What are they? Well, let's go all the way back to get bored. Where are we? The more complex ring structure ones, aren't they? Phenylalanine. Look at them all down here. Much harder to make. Takes much longer. That's why you ingest them. Except for this character, arginine. Arginine. Did you recall what arginine looks like? Okay, let's go back. There it is. Where is it? There it is there. A lot of carbons. 1, 2, 3 extra nitrogens. Whoa, hang on a sec. Didn't we say arginine was part of the urea cycle? What are you doing in the urea cycle? Managing nitrogens, yeah? Okay, there's some character that can deal with a lot of extra nitrogens. Let's have a look at why. Here's arginine. It takes 10 steps to make it, except if we got it from the urea cycle. Who can get it from the urea cycle? Well, you told me you're all past puberty, so you can. But if you're an adolescent, early life, growing, what are you doing? You're growing at a really rapid rate. If you've had kids, you know the midwife often comes around, weighs the baby every so days, whatever they are, it's every week or two. Gone past that point. As you get older, there you are against the door on this height and this height and this height. Your rate of growth is phenomenal. the demand for proteins, for laying down bone, muscle, gut, all those things to make you is very high in those early years. So arginine is in demand. So arginine is an essential amino acid for young children. Till you become an adult like us, and now you're old farts. Arginine is not such a requirement. Why? Because you can steal some from your urea cycle, which is where we're at here. You can see I've highlighted a couple of things, haven't I? Glucose, if I put a nitrogen on, modify a little bit, I can make an amino acid. I'm going to show you a couple of examples after we have a break. Now, I want to make a key point down in italics. What does it say? Many of these metabolic disorders, disruption to some of these enzymes, cause significant health issues. When you first come into the world, they stick you in the heel in this country, check you to see whether you have any disorders, because if you don't get to them straight away, many of them cause brain dysfunction. You would not be in this room if you had brain dysfunctions. You're in this room because they checked you early in this country, and then you got all the things you needed to be here. There are many who are less fortunate. Take advantage of it. Now, some of these are glucogenic, gluconeogenic. I'm going to talk about those as we go through the semester. That is glucogenic. Can I make an amino acid from a glucose carbon molecule? Should we have a break? Okay. Start back at 10 past.
If you can dim the lights a bit, it's just a bit hard seeing from up there.
I can try. Better? Just check. Hang on 1 second. Put this on mute in case it's...
It just seems a little bit overwhelming
All right. I promised the coordinator from the federal government in Canberra that I would show this slide. It's up on Views, I believe, and all their other information. They're claiming you can win $1,000 for signing up. They've been doing studies around the country, and they're doing, I think, 37 units this semester in this university. They're looking for 230-some thousand students to sign up to it, to demonstrate that your phones, much of you are glued to them all the time, might be useful in reminding you about how this unit operates. So they've asked me to write some things with them, that you get a text message and or your nominated person, whether that is, family member, friend or otherwise, but not me, reminding you that what's going on, what's happening, how are things working, what are the issues. That's to remind you that somebody cares, unlike some of your unit coordinators, trying to get you through units, particularly the federal government, because they want you to complete your degrees, as we all do. Okay, someone asked me to dim the lights a tad. Hopefully that's not too uncomfortable. I myself don't like too many high bright lights, particularly fluoros. Are we ready to go back to work? So we have some Maltesers down the front. They're going to roll them down the aisle like Jaffas.
Oh, no,
What's in that Malteser? Look at the nutrition label.
nothing much.
Sugar, lots of it. Fat, lots of it. So what are we going to do with sugar and fat right now? Are there any amino acids in there? Where's the nutrition label?
No. Oh, there's 6.3 grams of protein apparently
6.3 grams of protein in what?
per 100 grams.
Per 100 grams. Now I'm going to keep dropping hints like that all semester. Think about the units of measure. So one Malteser, is that going to weigh 100 grams? No, it's gone. How much does the packet weigh?
185 grams.
185, so you might have to adjust the volume, C1V1, C2V2. They're going to ask you that question in the lab. You'll have to do that all during the lab. You'll have to adjust concentrations to manage what you're doing. The protein nutrition label you bring in for your beverage, and someone's asking, can I bring in soya milk? That's derived from a plant, isn't it? Yep. It's not true milk, not derived from a mammary gland. Of course you can bring it in. But think about the nutrition label. What's the amount of protein? They're saying per 100 milligrams. So you need to think about your calculations. Well, you're using beverages, we're not using solids, are we? Sometimes they use percent per volume, volume per volume. We'll keep harping on that as we go through the semester as well. Must get your concentrations. Why? People die if you don't. Nurses in hospital are not getting the right concentrations and people are not being well, completely unwell. I use that term very loosely. Baby died recently because the wrong concentration of insulin was given to that individual. You must get concentrations. Screwed up my gene therapy when I was in CSIRO for a number of years until someone got the concentrations right. Not me personally, but around the world, it's happening. So you must get your calculations correct. You must have units and measure. Shall we go back to this work? Okay. I want to give you a couple of simple, to me, straightforward examples. And they are simple. You have to trust me on that. Don't have to trust me. Let's look at this first one. Non-essential amino acids from glucose. Let's go back one slide. What do we say? Glucose, how many carbons? 6. If I whack a nitrogen on somebody there, I might be able to make it into something like this purple box. What are those? The non-essentials, and this got essentials with an asterisk, depending on demand. I can make them, but if my demand is high, it's going to be harder to make them. That's why I gave you the arginine example. So even you, if you're lacking any of those amino acids, you're relying on glucose to make them. If you're trying to make things like protein, mass, babies, repair tissue, make antibodies, it's going to be a huge issue on your body, isn't it? Here's the first example. There's your glucose. Hopefully you remember that ring structure from last semester, or the linear structure. Keep in mind it's hydrogen bonding. 6 carbons in there to manage overall. What can you do? Well, lucky I had my flu shots, hey? Bless you. Man, that's scary. 4 amino acids from the glycolytic intermediates. What's the glycolytic intermediates? Glycolysis. We're going to keep working on glycolysis. I want you to look at this kind of grey box down here. There is glucose. There it is. Its structure, the beta-D glucose, the linear form, bless you. Goodness, if I get sneezed on, I'm going to die, I know. Glucose, right down 6 carbons. If I want to work with glucose to make it do something, what should I maybe do to it? Phosphorylate. I'm going to activate it. What do? Grab a phosphate from ATP often. I'm going to make phosphoglycerate. We'll come back to the details later. Don't be too impatient. I know how impatient you can be. But that's part of glycolysis. All that pathway, glucose, phosphoglycerate down to pyruvate, is glycolysis. You're just not seeing all the steps today. There I am making phosphoglycerate. If I go all the way down to pyruvate, I'm going to split those 6 carbons into 2 3 carbons. A few weeks away yet. When I split it, I've got a 3-carbon molecule. If I put a nitrogen on it, in this case, TA stands for transamination. I've grabbed the nitrogen from somebody else and stuck it on the 3 carbons of our pyruvate. What have I just made? You told me before, alanine. Phosphoglycerate, if I stick a nitrogen on it, ooh, lights are dimming. I didn't do that. If you put a nitrogen on, sorry? I'll try. You don't like it dark? Can't see. Is that better? You're welcome. Okay, we're awake again, aren't we? Phosphoglycerate, active carbon molecule, I can make serine. Think of its chemical structure. So nitrogen on there. From serine, I could make glycine. Serine, wasn't that a substrate? If I grabbed some sulfur from somewhere, I could make cysteine in this case. So there's a simple process of turning glucose into 1, 2, 3, 4 amino acids. What have you done? Stuck a nitrogen on them. In different places at different times. You've activated it first. Jot that down. Activated the glucose first. We're going to see you have to do that to make glycogen and other things later on in the semester. To do that, might require some ATP. That's our energy carrier. It also might require some electron-proton carriers to kind of G things up a little bit. We'll see those a lot during the semester. That's our first example. There's a few others in there. I'd like you to read the textbook. Last week I showed you some slides and said after each topic, after we finish amino acids, up on views in the amino acid module, you'll see some guiding questions. 10 questions, some multiple choice, some short answer. Each question will refer to a page or area in the textbook, the current textbook. I'm suggesting you read those pages. There's a lot of textbook to read, but just read those pages. Read around the topic area. If you read around it, you'll be more confident with the stuff I'm covering. What I'm covering is in the exam. You clear on that? Okay. So look at those guiding questions after each topic. When we finish amino acids, when we finish fats, when we finish carbohydrates, when we finish integration. They'll guide you to the nature of the questions I'm going to ask you, the depth of detail. And some of them might come back and bite you in the exam as well, in different forms. We finish with this one. Okay. This transamination reaction, look over on the right for a start. Don't write anything, just look at the right. Who do you see there? Alpha ketoglutarate. Write down alpha Kg. Let's go back a couple of slides. Where's alpha Kg? Critical in managing amino acid nitrogen either through transamination or the urea cycle. Where are you going? Is the nitrogen going to make some amino acids or is it going to get expelled going out through the urea cycle? Well, here it is. Alpha-ketoglutarate. Through this process of aminotransferase and this cofactor, I want you to circle PLP, pyridoxal phosphate. Might want to know that term later on. But many cofactors for enzymes are derived from vitamins. Not all, but many. So they are critical to help the enzyme work. Some of them, like magnesium, manganese, are ions that help enzymes work, particularly in the nucleus of the cell. Here is the process. So what am I doing? I'm using this cofactor derived from B6, PLP, this enzyme, aminotransferase. ASE, you know from last semester, don't you? FP&G. ASE on the end of anything means it's an enzyme. It has an active site. Well, what are you taking? I'm taking amino acids like aspartate or alanine, doesn't matter which. There's the nitrogen. That's what I love about Leninger. They actually colour code things so you can see where it's going. What do you do with that nitrogen with aminotransferase? You transfer it to alpha-ketoglutarate. What have you just made? Glutamate. Is everyone clear on that? The aminotransferase has transferred that purple nitrogen box to ketoglutarate to make glutamate. Simple reaction. What have you done in the process? Produce this thing called alpha-keto acid. What's that? Acid function, couple of carbons, which could be a molecule like pyruvate. And one you need to circle, OAA, oxaloacetate. You'll see it a few times later in the semester. Let's not panic about that one just yet. Lots of things to be patient on. But it's part of the citric acid cycle. So this process of transamination, we're moving nitrogen around. What do you need? An enzyme, cofactor. You need a substrate, which is the amino acid. You need some kind of carrier. This character is alpha-ketoglutarate. Go back to that nice diagram, the grey one. Where is it? Alpha-Kg. Are we done with this one? No? OAA stands for oxaloacetate. And I'll keep coming back to these acronyms. You think there's going to be lots in this unit, you wait till you do molecular biology, biochemistry, cell biology. Sorry? Transamination. You're aminating somebody. You're moving an amine function from one character to another character, which I believe was just back on this slide. See it there?
Oxaloacetate.
Sorry?
What is oxaloacetate?
Oxaloacetate, yes. So think of alpha-ketoglutarate as alpha-Kg. What is it? 1, 2, 3, 4, 5 amino acids, 5 carbons, I should say. An acid function. An acid function, what do? Stick a nitrogen up there where that ketone is. You've now kept the nitrogen away from being toxic. You don't want free amino groups. And just while I'm on those free amino groups, it's about concentration. Too many free nitrogens, ammonia, is toxic. You have a little bit in your blood, tiny amounts in your cells. It's about concentration. Too much water will kill you. Think about concentration range. We done with this one? I'm comfortable with that. Transamination, moving nitrogen around. Cofactor, substrate, product. Let's get to the next one. Let's see what glutamate does. Where's glutamate? Go back here. There it is. Nitrogen, few carbons, acid function. What are you doing? Making glutarate. Here's alpha-Kg again. There's PLP, transamination. On the left side, I just showed you that in the last slide. Transamination, what have you made? Glutamate. Let me go back one slide. Can you let me go back one slide? Is that a yes or a no? No? Yes? There it is there. That process, alpha-ketoglutarate, glutamate. Just circle that in your minds. Go to the next slide. There it is there. Alpha-ketoglutarate, transamination to glutamate. What have you done here now? Well, they've got this thing, glutamate, with the alpha-keto acids. You can make alpha-ketoglutarate. You can recycle the process. So your, bless you, glutamate might only be a transient molecule. Bless you. The other character that can help is this thing called GDH, glutamate dehydrogenase. What's that doing? Well, same kind of thing, but it doesn't have the cofactor. There it is up here, GDH. So our transamination reaction is moving our nitrogen functions around, but it's using a bit of ATP. It requires energy. Anytime you move something, it usually costs you energy. Everything's at a cost. What are you trying to do? Well, as I said, trying to make sure ammonia is not free in high concentrations, which becomes toxic to your cells. What are you going to do with that particular nitrogen? Well, at some point next week, we're going to turn that into urea. At some point, that's going to go to your bladder. At some point, that's going to go into the porcelain and be dealt with by somebody else. But while you're doing that, you're going to turn those carbons into other things like glucose, maybe fat, depending on, just write it down, supply and demand. If you have sufficient glucose, you'll turn the carbons into fat. If you don't have enough glucose, you'll turn some of those carbons from the amine group, the amino acids, into glucose. Now, some of these characters end up in the circulation, some end up in the kidney and off into the porcelain as well, depending on how much is available in your cells and tissues. Now, as I said, last half of the semester, we're going to focus on that. What regulates how much goes through your body, particularly in your blood circulation and lymph. So this glutamate, as I have in italics, is critical for amino acid synthesis and degradation. It's key. Let me go back to that slide that's important. That's key. That key pathway. Who's the key enzyme? Transaminase. Who's the key cofactor? PLP. Who else can do it? GDH. Those are the things you should be starting to jot down as we go. Are we done with this one? Okay. Let's look at a couple of characters like serine and glycine. Now cast your eyes on the right-hand panel. Work down. Do you recognise what path that is for a start? Glycolysis. There it is. Glucose. You've activated it, put a phosphate on it. You have a number of steps we'll come back to. There's phosphoglycerate. Phosphoglycerate, if you were a plant or a bug, you could make erythrose 4-phosphate, 4-carbon molecule, do other things with it. We generally don't. 3-phosphoglycerate, we could make serine. Get to that in a moment. From serine to make glycine or cysteine. A few more steps, we could make phosphoenol pyruvate. I want you to write down PEP. You're going to do a will assay, use it as a template, in a few weeks' time called PEP will assay. Phosphoenol pyruvate. Phospho tells you it's activated. Phosphoenol pyruvate could be used to make things like tryptophan, phenylalanine, tyrosine. Do you make those? Remember, there are a whole lot of steps to do that. Who makes those? Generally other species like plants and bugs. I kept on going. I can end up with pyruvate. We just saw we could take put a nitrogen on there. We can make alanine, valine, leucine, isoleucine. I'm going to come back to the red box. Let's go back up here to the glucose 6-phosphate. Glucose 6-phosphate, take off a carbon, ribose, 5-carbon sugar. Who makes that? Plants and bugs. Do we do that? Not so much. What do? They make histidine. And that's an important amino acid. It can be used in biotechnology. We'll talk a bit about that maybe. If I get down to pyruvate, I might enter this thing, the citric acid cycle. The citric acid cycle has a number of steps. Now, this is where you break carbon bonds. Usually in the first half of this cycle, and here's one of those key carbon bonds, alpha-ketoglutarate. Write it down, alpha-Kg. It's an intermediate in the citric acid cycle. And right now you're thinking, what the hell does that matter? Well, when you break carbon bonds, what do you generate? Not flames. Energy, electrons and protons. You can make more ATP. So if I'm using alpha-Kg to do other things, I'm slowing down my rate of energy production. Supply and demand, the usual issues. I run around that cycle, a few more breaking of bonds, a few more electrons. I get around to this thing. What's this thing's acronym? OAA. There it is, part of the citric acid. It's an intermediate. If I use OAA for something, what do I do to my cycle? If I bleed it off, I can't finish the cycle, I slow it down. Anytime I'm managing excess nitrogen, I slow the production of energy down in cells. Keep that in mind. High protein diet does what to you? High protein diet, sitting here looking at me, what are you doing with it? Creating fat. What are you doing with all that nitrogen? Making urea. What are you doing to cells trying to make energy? Slowing them down. So you become almost more lethargic, don't you? Because you have less energy to do the work. It's a vicious circle, isn't it? So don't sit there making fat. From these two characters, you can make other amino acids. So if I'm deprived of glutamate, glutamine, proline, arginine, I bleed out of that pathway. I slow energy production down. OAA can be used to make aspartate, asparagine, methionine, threonine, lysine. But if I bleed OAA out, I slow energy production down. There's a cost. If I have to make amino acids, my energy production is slower. I can't make as many things. That's my balancing act. Energy production for work or building blocks. Which one? I have to manage that. So that's why you ingest. Let's have a look at these red dots. Can I move to the next one? Okay. Here's the first example of a grunt biochemical pathway. Up to now, I've been taking you in the shallow end, dipping your toes, getting you used to what's going on. We're starting with an activated 3-carbon molecule. There's our phosphate. What do we need? A dehydrogenase called a phosphoglycerate dehydrogenase. Who's our helper? NAD+. What does NAD+ do? It grabs electrons and protons and helps activate things. I've just now made 3-phosphodihydroxypyruvate. There it is, the pyruvate molecule, the phosphate on it. It's active. I can do a few things with that. I'm not going to tell you what they are right now. One of those things is if I grab glutamate, what's on glutamate that we saw in green or purple recently? Glutamate carries what for you? Well, if it's green in that box there and it's green in that box there, what's it likely to be? Nitrogen. Glutamate is your nitrogen carrier. It protects you from ammonia. Here it is as glutamate donating a nitrogen to this thing, 3-phosphoserine. What have you produced? Write down alpha Kg. Phosphoserine phosphatase. Okay, let's talk about kinases and phosphatase. What does a kinase do? It phosphorylates somebody. What does a phosphatase do? Takes the phosphate off. That's as simple as that. We're going to see that all semester. So if I see a phosphatase, I know somebody's had the phosphate ripped off them. What did I just do to serine? I took the phosphate off. Here it is, 3-phosphoserine. I've just made serine. Now what can I do? I can use this cofactor, folate. Most women have had children, have heard of folate. Keep coming back to these kinds of issues. PLP, we just saw a moment ago, didn't we? Pyridoxal L-phosphate, derived from vitamin B6. What do we do here with this enzyme, the transferase? A transferase, a TA. What have I done? Move things around. What have I made? This case, glycine. There you are, making simple amino acids, starting with what? What did you start with up above here? A glucose molecule. You activated it first, put the nitrogen on it from our friend here, our compatriot, our donor, glutamate. You went all the way down and made 2 different amino acids, serine and glycine. Serine first, then glycine. If I have a high demand for serine, it's going to be used a lot in a lot of amino acids, proteins, it's functional activity. I'm not likely to make as much glycine, am I? But if I don't need as much serine, I could potentially make more glycine, couldn't I? That's important for your project. It's important for what you do in the lab. That standard is what? What's the standard we're going to give you to use to measure your beverage? What was its acronym? BSA, bovine serum albumin, about 50-odd, 52 kilodaltons. What is the composition of its amino acid sequence? Do you know? Should you know? I would find out. What's the amino acid sequence of BSA? What's the ratio? How many alanines? How many serines? How many threonines? Is it disulfide bonded? Does your red dye measure something unique in that molecule? Does it measure the individual amino acids? These are all hints for your lab activity. They'll be asking you those questions and you'll get a nice round number or a nice straight number, depending on whether you've done your homework. Is that kind of clear? Think about that protein. Are we done with this one? So the concept is activate it, mess with it, move nitrogens, and now you've got an amino acid. If I remove the nitrogen, I manage the urea. Bless you. Now you've been going how long since lunchtime? You had lunch at, say, 12, 1 o'clock. What's the time now? 5:30. Around 4 hours. You are now going into the fasted state. What do you see? Gut, no incoming. No bacon, no steak, no tofu, nothing. What's happening to you? Well, you're sitting there, and because you're only using the odd finger, maybe trying to keep the eyelids open, breathing hopefully, heart's still pumping hopefully, so there's some work going on. But what you're doing is if you're not using some of those muscles, you're tolling off the nitrogen. You might have to recycle them. Alanine we're going to see in a moment. Glutamine we saw as a carrier of nitrogen, didn't we? It can do it outside the cell as well. So can alanine. Glutamine can help manage kidney activity. Kidney can help make things like serine alanine, which is kind of unusual. Kidney does a couple of weird things, not just filter your waste products, but it's going to get rid of the ammonia. The glutamine can go via the gut. Now, this arrow doesn't mean alanine goes into the gut lumen and then back into you. It just means it passes through those epithelial cells. They can use it because they're kind of benevolent. Their job is to feed you, pass things through into your blood or your lymphatics. Where does it end up? The master controller, the liver. Supply demand. They have to respond, those hepatocytes. What are they doing? Well, if you've got excess nitrogen, you make urine via urea. That's the urea cycle. Where does the urea cycle work? In the liver. Right down in the liver. Not muscle, not bone, not fat. In the liver is where you make your urea. So you have to get all those nitrogen back there. In the liver, you'll make glucose. In the liver, you'll store some glucose as glycogen. In the liver, you'll make fat. In the liver, you'll make these characters as well, ketone bodies. Now, we'll see those later in the semester. They're happening the more you fast and when your blood sugar keeps getting lower and lower, not zero, but lower. Brain's not working too well. Liver's saying, I can't make glucose quickly enough. You start making ketone bodies. I'm going to save them till the end of the semester. They're very simple molecules, but I want you to jot down, your brain uses glucose, not fat, not amino acids. It builds things with them, but doesn't use them to make energy. But when you're in a high demand and you're low in blood glucose, ketone bodies can help your brain work, but not very well. You can smell them on your breath if you get in a diabetic state. I'll discuss that later on as well. So the liver's critical. I underline in italics there all. The liver manages everything. That's why you need to look after it. Don't abuse it with drugs and alcohol, even though it's one of those organs that can come back from almost death to recover. Its recovery is actually quite good, but you must look after it. Sorry?
Your brain uses what?
Your brain uses glucose. That's what your brain uses. It doesn't use fat to make energy. It doesn't use amino acids. But when your blood glucose gets really low, it starts using ketone bodies. But those ketone bodies don't help your brain work very well. You make lots of bad decisions when you're using ketone bodies in your brain. Comfortable with that? Is everybody else right? That's why you maintain your blood sugar within a range, the homeostatic range. Yes, ma'am?
You know how you're saying your brain doesn't use ketones as well?
They do when it's in high demand and your brain is running out of glucose. Yeah?
Because I was wondering what the effect of ketosis diet would have on the brain function.
Yeah, we can discuss that in more detail as we go through the semester. The question was ketosis diets. You know, those are potentially dangerous because you're pushing your body not to make as much glucose, but to use ketone bodies. When you use ketone bodies, the brain doesn't work as well. You've seen those ads a few years ago where the guy in the pub, you know, his brain's sparking away, a few more beers, doesn't spark too well. That's pretty much how it is. You know, your brain uses glucose. Ketone bodies are very simple carbons, but it doesn't give you the way neuronal function as well, because in your brain you're using a lot of energy to pump sodium out of neuronal cells to cause that action potential, which I can cover in another unit later on. But yeah, that is critical. What you eat, how you eat, what your brain does is important. And your liver, you might want to jot down, is so busy trying to maintain blood glucose for your brain, it might start neglecting other activities, because that's its major role. And that's why we see its major activities are the key enzymes for all the pathways are there. You can see they're interlinked. Let's go all the way back to this slide.
So what is it try to control?
Just one sec. So this slide, remember I said this is the liver? These are all the key pathways. I'm sorry, what was that bit?
When you think that the liver controls what again?
The liver controls all the pathways. Incoming fats, outgoing fats, incoming glucose, outgoing glucose, incoming amino acids, outgoing amino acids. Everything, basically. The liver also detoxifies. How many of you eat pate? Yeah, I used to when I was a kid because when I grew up on a farm, the liver from animals was pretty clean. These days I wouldn't touch it. Too many toxins are accumulated in the liver. Unless those animals that you're ingesting their livers, their pate, is from an organic-based diet. Your liver accumulates things, unfortunately. Can I move to the next one? Yep. All righty. Alanine. There's glucose. What do you have to do to use it to do some work? Phosphorylate it. There it is over on the left. What do you run down to? If you run to pyruvate, you've made a 3-carbon molecule. If I want to turn that to alanine, what do I got to do? Transaminate it. Put a nitrogen on it. That alanine I've just made, I can build something with it, like BSA if I was a cow, or a bull, or a steer, whatever the case was. But that alanine is also, I've hinted before, a nitrogen carrier. Let's go back a slide. There it is. There's my muscle, I'm not using it, or I use it really hard. I can recycle alanine. 3 carbons and a nitrogen. Here it is here, 3 carbons and nitrogen. You notice that double-ended arrow. I need you to notice those all through semester. Some arrows are one way. One's an enzymic step, only that direction. Others are two-way, depending on supply and demand. In this case, alanine, if I have lots of it not being used, I can turn it into pyruvate. If I can turn it into pyruvate, that dashed line, I want you to write down gluconeogenesis. I'm making fat from amino acids. I'm making sugar as well. In this case, I'm making glucose. Alanine, as I have in italics, is a major gluconeogenic. You use an amino acid to make glucose, particularly when your blood glucose levels are low. That's what we have over here in the dotted red pathway. Alanine, pyruvate, pyruvate, alanine. But if I mess up glycolysis, I slow energy production down. Always there's a cost. Now this pathway, as I hinted a moment ago, often runs between your muscle and your liver and your muscle. You build up protein in your muscle, you need alanine. You might want to jot down alanine as a common amino acid and very abundant in most proteins, particularly things like actin and myosin in these muscle groups. Can I move to the next one? Okay. This cycle you need to know, there's going to be lots of cycles. Well, not that many. This is the alanine cycle. There you are, working away in the gym. Catch me in the gym anytime. Working away in the gym, what are you doing? You're encouraging activation of transcriptional factors to lay down more actin and myosin, build up protein mass. But what happens if you work that muscle really hard? You start running out of glucose to do the work. You start this process here of recycling it. You'll see pyruvate is going to be used to make energy as ATP and the like. But through this pathway now, pyruvate's going to have its nitrogen put on to make alanine in muscle. In that muscle now, we're going to see the alanine disappear and head off to your liver. Alanine gets down to your liver via the bloodstream. In the liver makes a decision. The problem now, children, in the gym, or adults, is that you're working so hard, we're low on blood glucose. What do we need to do? Turn that 3-carbon nitrogen compound of alanine into glucose. Here we go. Take the nitrogen off. Who does that? Our nitrogen carrier. There it is, glutamate. What do you do with that? Send it off to the bladder. What do you do with the pyruvate? Gluconeogenesis, you've just made glucose. What does your liver do? Dumps out glucose into your circulation. What's it trying to do? Keep your brain working. Whatever you're doing in the gym, you might want to stop because this thing's not working too well. Does that kind of make sense? The rate of turnover is critical. Now, this is a simple one, the alanine cycle. There's going to be one later on called the Cori cycle, which will look similar, but it's not the same. And why I'm highlighting that now, because I often ask this question in the exam. Explain the alanine cycle. A couple of multiple choice. Then there'll be a Cori cycle one. They look similar, but they're not the same. What does alanine do? Recycles alanine from muscle to liver to glucose back to the circulation, particularly under high demand. We're done with this one. Is that a yes? Anybody else right? So you're going to get in the gym after this? Should you go down to the gym while you've been fasting for 4 hours? Think about what your body has to go through. Should you go down to the gym straight after you've had a big meal? You're busy trying to digest and absorb. Think about what your body's going through. That'll help you in this unit, because then you'll be able to explain those things in the short answer questions. Can I move to the next one? Okay. This process of turnover, I've given you just a handful of examples, very simple ones, to get you an idea of how you turn over, either synthesize or degrade these amino acids. Think of them as carbon skeletons, maybe the odd sulfur, a few other things on them, but definitely with a nitrogen or 2, maybe 3. I have to manage that nitrogen so it's not toxic. If I'm going to build it, I have to activate it. I put a phosphate on it. I'm going to do something with those carbons. I'll turn it into sugar or fat, or make it to some other molecule. So these carbon skeletons are good to make some things. Now, ketone bodies, I want you to jot down, there because you don't go around the cycle fast enough, the citric acid cycle, so you shoot ketone bodies out. It speeds up the process of getting simple carbons for the brain. This is a balancing act. So the fate of these carbons from our amino acids is similar to what happens to your carbohydrates and fats, because aren't they carbon-based molecules? Should I use some of those to make things like sugar turn into amino acids and vice versa, depending on what your diet is? If you're on a ketosis-based diet, if you're on a high-protein diet, a high-sugar diet, a high-fat diet, it pushes the enzymes a different way. And let's think about that. That's critical. All the pathways I've shown you today and any others, if I'm just on a protein diet, means the enzymes to manage proteins like alpha-ketoglutarate, PLP, transamination, the urea cycle, what do they need to do? You need more of them because your diet is just protein. I've heard of someone who's just had protein diets, now they have colon cancer. Those people just on fat diets, that'd be pretty dangerous. Those people just on carbohydrate diets, the enzymes involved have to increase to manage that to try and balance out the rest of the things you don't ingest. That's why you have disease states. I knew a guy when I was an undergrad who was a mathematician going through his undergrad, pure and applied maths, but he only ate Vegemite on toast. No butter, just Vegemite on white toast. 6, 7, a dozen slices for breakfast, 6, 7, a dozen slices for lunch, and maybe 10 or 15 slices for dinner. That's it. Nothing else. No chips, no fish, no vegetables, nothing. He didn't look particularly healthy, but he was a good mathematician. That diet meant his metabolism was completely shifted to what? What's in bread, mostly? Carbohydrates. What's in Vegemite, mostly? Apart from the salt. some minerals, a few amino acids, so he could probably survive, but it meant all of his metabolism was shifted to just that diet, which is why you want a balanced diet. You done with this one? Is that a yes? Okay, let's summarise that. At level 2, I generally try to summarise the day's work in a slide or so, to kind of bring it back to earth, remind you of the things we've covered. Amino acid biosynthesis, that's synthesis and degradation. Now, I have not taken you through all the complex ones. Is there any point? Well, there's just more pathways, more steps, more cofactors. And the other thing you want to thank me for now and at the end of this semester is I'm not showing you any delta Gs. There's no change in free energies. We had to do all that by memory. You don't have to do any of that because delta Gs happen in a tube. In your cells, it's too variable, as my research and my students know full well. You don't have to do delta G. So when you see that in the textbook, ignore it for this unit. Is that kind of clear? But for this, you need to know glycolysis. You're starting to understand glycolysis, gluconeogenesis, starting to understand the citric acid cycle, its importance in managing these pathways. I'm not covering bacteria and plants. Each of those is specific for their activities. You've realised now, hopefully, the amino acids are building blocks, but also potential energy sources, but also potential messengers, extracellular messengers. They can sometimes be cofactors. What you eat, are you a carnivore, an omnivore? My daughter calls herself a meatitarian. Hopefully she has a balanced diet as well. You're eating just vegetables. How many proteins in vegetables? Any? There are some, but it depends on the vegetable you eat, doesn't it? It also depends on the time of year, what season, how much protein they're putting in to that particular vegetable you're eating, or fruit or salad. You're trying to manage that. Are you a herbivore? Some of you are. But your metabolism is designed as an omnivore. You're designed to be able to eat meat, protein-based and plant-based materials. That's what you're designed to do. What else? Had enough for one day? Is that a yes?
Yes.
Let's go.
It's happening now. We practiced last week, so anybody who says they can't get on, what was the acronym? NMP. When you're ready to go, I'll tell you the password. Wait, no, not ready. Sorry? Just make sure they're on there. Was it?
No.
NMP, yeah, that's right. No, it's not NMP. Are we ready for the password? Are we there yet? Okay, it's all caps. HDG. HDG. Now, last week we did a week practice, as in WAK. Wasn't that challenging? I'd encourage you to read carefully. I'd encourage you to collaborate, cavort, discuss, whatever you need to do, but talk to your associates. Find a brains trust to help you out. There is no excuse that you should not be getting full marks for this, is there? When you are done, we are done. I'll see you next week. We're in, I assume. Got questions? You're in?
Oh, yeah.
Yep, cool. I can't hear you, too quiet.
Can you cheat?
Question was, can you cheat? I wouldn't call the word cheating, I'd call collaborating. You'll note we finished early as I promised we would. Start on time, finish 10 minutes early. Thursday, 1 o'clock.